have to make a gene, and we have to use the promoter, riboson binding site, start code, lipase, wash enzyme, start code, and the terminator are being set together as a gene. The promoter is being inserted first so the DNA string can split up. Then there is the ribosome binding site, which make the mRNA bind itself to the ribosome. During translation, the ribosome starts reading the mRNA and starts building the enzyme that when it meets the start codon. Lipase is the code for the amino acids composition to the enzyme that is wanted in the end. The stop codon is where the mRNA stops. The terminator is the last thing, and the strings get cut off. And then the gene has to be altered. The made gene and the yeast needs to be electroshocked so the cells can absorb the DNA easier.
The proteosynthesis takes place in the nucleus, and the DNA, which is double-stranded strings, opens up. The enzymes and proteins open up the strings near the promoter, and when the enzymes bind to it, the RNA binds to the DNA's one strand, where the RNA polymerase reads the DNA it had selected, and when the polymerase is at the end, it has made a mRNA string made by the base pairing principle. When the RNA polymerase reaches the terminator string, it cuts it off again, and the mRNA strand is being transferred to the ribosome, where the tRNA reaches the ribosome and the amino acids. The tRNA delivers its amino acids, and each time the 3 bases have been read, there will be put on a new one, and there will be formed a long chain with amino acids, and the chain will fold together by the help from an enzyme. And that's what happens in the electro.
Then we have to have the genetically modified cells in the petri plate where there are antibiotics, and we do this so it will kill the cells who wasn't getting genetically. The genetically modified cells need to stay in the heating cabinet for 24 hours so the cells can multiply. Then the cells colony transfers to the test tube, which contains growth media and antibiotics. It needs to be set in the heating cabinet for 24 hours again. The fermentator's pH value and temperature are being adjusted so it can produce as much as possible washing enzyme.
The gene needs to be transferred to the fermenter, where the genetically modified yeast can grow under controlled conditions and produce as much protein as possible. The pH value is being set at 5.0 and the temperature at 30 degrees, which are the best conditions for yeast, and that is where the cells will develop fastest. The chromatography equipment makes protein purification where disrupting substances are being removed. The finished product is being collected in a cannula and transferred to 6 test tubes in different dilutions. At least the gene needs to be tested. The best solution is to use the enzyme with the dilution 1:1 and a temperature at 90 degrees. But because it's not environmentally acceptable to use that high temperature, it's best to use the product in 20 degrees. There's a big difference between between washing the shirt with or without the product.